मैम अभी हमारे यहाँ लाइट गई है जस्ट,
रीना
तो
मिश्रा
मैं
मैडम।
डेटा से ही
मैडम आप
बोलूंगी।
मोबाइल से ज्वाइन कर, अभी
हाँ
आप किस
जी,
डिवाइस से ज्वाइन है?
मैं मोबाइल से ज्वाइन हूँ
ठीक है
सर
ना मैम, मैं
मोबाइल,
यहाँ से
और
शेयर
अभी
करती हूँ।
लाइट गई है तो।
ठीक है, नो नो
हाँ
इश्यूज।
ठीक है
आप वीडियो ऑफ ही रखिए। और
जी।
वॉइस तो आपका अभी आ रहा है क्लियर। वी विल स्टार्ट द
हाँ
सेशन। आई विल शेयर द पीपीटी।
यस।
एंड आई रिक्वेस्ट ऑल द पार्टिसिपेंट जो भी प्रेजेंट करेंगे आज से आगे भी, कि प्लीज स्टिक टू द टाइम ऑफ 10 मिनट्स। 8 मिनट में कंप्लीट करने की कोशिश करिए। आई नो आप लोगों ने बहुत सारा वर्क किया है, बहुत सारी चीज़ें ऐड की है पीपीटी में, सबको बताने की भी आपकी इच्छा है। बट एज फार एज टाइम इज कंसर्न, प्लीज स्टिक टू द टाइम ऑफ 8 to 10 minutes। इन शॉर्ट यू हैव टू एक्सप्लेन एवरीथिंग।
ओके मैडम।
रीना मॅडम पीपीटी दिसतंय का?
हो
होय
दिसतंय
मॅडम,
मॅडम, पण
दिसतंय
फुल
येस।
दिसतंय.
स्क्रीन नाही दिसत.
तुमचं पीडीएफ पाठवलं मॅडम, दिस इज अ पीडीएफ फाईल.
हा पीडीएफच
येस,
आहे माझं.
ठीक आहे, प्लीज स्टार्ट.
येस. Good afternoon, sir. Good afternoon, madam. And good afternoon to all the teacher participants. I, Dr. Reena Meshram, Assistant Professor in Botany from Nabira Mahavidyalaya, Katol. And today's my presentation topic is Survey on the Application of Pesticides and its Consequences on Farmers' Health in Nearby Places of Katol. Ma'am, please next slide. Just a minute. These are some of the brief contents of my presentation. Please next slide, ma'am. Domestication of useful plants in the form of agricultural practices is perhaps the one of the most important event that has changed the livelihood pattern of human being from foragers, hunters, and agriculture to gatherers to agriculturists. And from this, we started forming colonies. Okay? When everything was perfectly right, then why there was a need of use of pesticides? The answer is Second World War. It is responsible for the use of pesticides because there is a scarcity of food and we need more food as compared to that of the population at that time. And Second World War duration is during 1960. Okay. uh, During Green Revolution there has been introduction of high yielding varieties which are nothing but the HYV, which require more fertilizers, more pesticides, and more water. It is still not clear when this random use of pesticides and fertilizers of Green Revolution era converted into mandatory activity or component of modern agriculture. Please next slide, ma'am. These pesticides have their inherent effects. It affects air, water, soil, and ultimately circulates in food chain. As we are at the higher tropic level, we are suffering from several diseases like abnormalities in eyes, respiratory problems, skin problems, cardiovascular diseases, kidney related issues, and cancer. Please next. By extensive review and from experience of neighborhood farmers, we are decided to conduct survey in nearby villages of Katol, as I am from Katol, on consequences of pesticides on farmers' health because he has a first-hand experience of pesticides. We are directly or indirectly come in contact with the pesticide, but our farmer, है ना, he is actually first-hand experience of the pesticide. So these are some of the objectives. Please ma'am, next slide. Objectives of our survey: to obtain information about the use of pesticide in farmlands, to gather knowledge how extensively they apply pesticides, to acquire information about precautions taken during application of pesticides. These all are related with the farmers only. To gather information about health issues after pesticide applications. uh, Please ma'am, next slide. Bishnur, which is around This is the site of study. Bishnur, which is around 25 kilometers from Katol, was selected as site of study as majority of the population in Bishnur is engaged in farming. Its population is around 5,000. A total 100 farmers have having various levels of educational qualifications were randomly selected for collecting the required information for this study. The respondents were not pre-informed so as to avoid biased responses and to gain actual insights of the farming practices. However, at the time of the interview, the farmers were apprised with the aim of the study. Interviews were conducted in local language and a verbal consent was taken from all the participants. Data was collected by means of structured questionnaire, one-to-one formal interviews. Please, ma'am, next slide. Overall data in this survey was classified into three sections: personal, about the farmers; pesticides use and management practices; protective measures; and health issues. Please, ma'am, next slide. Now we come to the results and discussion as we have very short time for all these things. So here in social demographic factor, first age and gender. The respondents belong to different age groups, that is from 22 to 75 years, with 48 years age groups farmers were more. Gender wise, only male respondents were there in the farmers, and actually my student, she went to the farmland and she actually collect all this data from the farmlands only. Please, second slide, ma'am, next slide. When we talk about the economic status of our farmers in Bishnur, here we found that the income group statistics presented a mix of all income groups. All farmers have their own land, but there is a difference in their annual incomes that is ranges from 30,000 to 2 lakhs. 59% farmers belong to low annual group, that is 30,000 to 50,000. Only 6% farmers belong to high income group, that is from 90,000 to 2 lakhs, which is again very low. But in our survey, in and among the 100 farmers, this is the highest income group we found. Now the qualification of the farmers, which is one of the main factor which overall develop the power of thinking analysis. We can think like that because we are the teachers and we are always thinking in that context only. So among 100 respondents, approximately 15% respondents were below 10th standard. 30 farmers completed their SSC education. 30% are HSSC passed, matlab 12th pass. 20% are graduated, and only 5% respondents different pesticides used by farmers. It has been observed that 48% farmers use weedicides and 41%, ma'am, next slide please. 41% farmers use insecticide only, few use fungicide and rodenticides. Um, Next slide please. Just a minute. It is not clearly visible in the
Yes, ma'am, it is a table of result and
now.
discussion.
In this slide, we ask some questions which are not mentioned in the form of the graph. The questions were duration of applying pesticides. We asked these questions to the farmers, and they respond that minimum 3 months to 6 months. These much pesticides they have used in the farmlands, and most of the crops they
हेलो।
taken are soybean, cotton,
थोड़ा
etc.
कनेक्टिविटी का इश्यू होगा।
Most of the
रीना
farmers
मैडम,
they
हेलो।
taken the crop are soybean and cotton. We can think about two crops only because it is citrus plant is orchard is there in Katul belt, but these two crops were we found in Bishnur place. Mixed different brands of pesticides when we asked them that they use the single pesticide or they mix the different brands of pesticides, then 92% farmers said that they use mixed brands. I am audible?
यस मैडम,
Yeah.
अभी बीच में
हेलो,
आवाज
हाँ मैम
यस
बोलिए।
यस।
हेलो,
हाँ, हेलो
हेलो,
हेलो। हेलो मैडम, मेरी आवाज आ रही है?
हेलो, हेलो, हेलो।
मैडम यू आर ऑडिबल, प्लीज कंटिन्यू।
Hello.
शिट,
हेलो रीना मैम,
एक
यू आर
मिनट
ऑडिबल।
Yes, you are audible, ma'am.
यू आर
हेलो, एम
ऑडिबल।
आई ऑडिबल? हेलो,
यस यस
हेलो।
यस। शायद उनको अपनी आवाज नहीं जा रही है।
ऑडिबल है मैम।
हाँ, हाँ, हाँ। तो
किसी के पास मैडम का नंबर होगा तो उन्हें कॉल करके बताइए कंटिन्यू करने के लिए।
when we ask that how much
या
stress सभी को आ रही है?
यस यस मैम।
Okay,
नो मैडम,
okay,
इट
ma'am.
इज ब्रेकिंग।
How much stress do you think you are exposed to while using
वॉइस
pesticides?
इंटरप्ट
Then
हो रहा है थोड़ा।
only 4% farmers said that there is no risk to use the pesticides. Hello. And 90, hello, am I audible? Madam, I'm audible?
हाँ यस।
Yes, yes.
मैडम डिस्कनेक्ट हो गए हैं, ठीक है। एनीवेज विल कंटिन्यू हर प्रेजेंटेशन लेटर। She was explaining in a nice way actually. Dr. Mayank sir, are you there? Dr. Mayank?
यस मैडम, आई एम हियर।
Yeah,
आई एम हियर।
yeah. Sir, please be ready. मैडम ने तो अभी ज्वाइन किया है, लेट्स सी उनकी वॉइस आती है क्या, अदरवाइज फिर आप कंटिन्यू करिए।
यस मैडम, आई एम रेडी एंड प्लीज गिव मी द परमिशन टू शेयर माय स्क्रीन मैडम
यस यस, जस्ट अ मिनट। हाँ, मैडम ने ज्वाइन किया
फॉर
है।
प्रेजेंटेशन।
या
थैंक यू
Ma'am, am I
जी।
audible, ma'am?
हाँ, नाउ यू आर ऑडिबल रीना मैम, जस्ट आई एम शेयरिंग वंस अगेन।
Yeah.
You just continue from result and discussion.
Yeah, yeah.
Yes, continue
Um,
please.
yeah. Then the next question was, can this usage of pesticide cause any kind of long-term or short-term health effect on you? Then 10% say no effect on the farmers, and 90% farmers said that they have effect or they have the visual effects on their body. Then the next question was, how long duration after applying pesticide do you re-enter the field? मतलब आज पेस्टिसाइड डालने के बाद आप कितने दिन बाद फील्ड को रीविजिट करते हैं? देन दे सेड दैट 12 to 16 आवर्स, एंड इसका जो मीन हमने निकाला वो 14.8 आवर्स जो है वो हमने लगभग निकाला है। प्लीज मैम नेक्स्ट स्लाइड।
Ma'am, the
देन
slide,
ऑल द
yeah.
फार्मर्स all the farmers mentioned suffering from at least one type of ailment during the course of pesticide usage. That is skin irritation 38%, nausea 7%, fatigue 9%, eye irritation 8%, and respiratory problems 18% were the most common symptoms which mostly found in the farmers. Just a minute, ma'am.
Ma'am, which slide you are?
Yeah, I'm telling you. Actually,
Yeah,
I am lag behind. Yeah. Now, result and discussion पे आइए. Most of the farmers among the 100.
yeah. Okay, continue.
Yeah. Most of the farmers among the 100 farmers were it is what I can say, the brief. 100 farmers were approximately 45-year-old males with average education and belonged to the low income group. The majority of the opinion that they were aware of the health hazards of pesticide use and suffered from symptoms like redness of eyes, nausea, fatigue, respiratory problems, and nervous problems. Surprisingly, the opinion was almost equally divided on whether they perceived pesticides to be cause of their health problems, and major percentage did not avail of the medical help even. Most of the farmer respondent that did not receive any training in pesticide handling and use partial personal protective equipments during working hours, but most of the farmers lying with us that they use the mask, hand gloves, etc., etc. But we found that they never use mask, they never use gloves, they never use PPE kits, and nothing. They all lie with us that they use all such things. The farmers did not read the prescription written on the bottles and haphazardly utilize the pesticides. They wash the farming tools in the nearby water body, and their cattle drink the same water, which may lead to various health hazards in human because we drink cow's milk. This is the indirect consumption of pesticide. We found variation in their opinion in case of use of pesticide in farmland. Some said that pesticides are necessary for the high production of crops, while others said that it has deteriorate the quality of soil and environment too. We got the mix. What we can say, the response is mixed. These observations clearly highlight the urgency of improving the awareness education towards the short and long term effect of pesticide use. Now, when we come to the conclusions, what we conclude from this short survey, that it is well known that with the emergence of the green revolution, fertilizers, pesticides, hybrid varieties are launched in India. It may be the requirement of that era, but when these come into the practice, nobody realized fertilizers and pesticides became catchword for enhancement of productivity of crops. Farmers have blind faith on pesticides without knowing their effects. Even sometimes, when there is no need of treatments to crop, they also spray pesticide, and that too very randomly, with the hope of high crop production. This is the general scenario which was observed during the study. We found that a sizable population farmers were severely affected by pesticide exposure, and health hazards range from mild skin irritation to other severe diseases. The following measures should be implemented to stop pesticide poisoning mediated health problem. That is, first, the farmer should have safety measures such as at least two sets of PPE, one of which can be worn when other is being sterilized. Proper sterilization procedures must be made available in farm to facilitate the effective cleanup of pesticide residues. In case of autoclave, they can use the pressure cooker also. Regular health checkups must be mandatory to identify any symptoms of pesticide poisoning in the early stages, thereby ensuring good health of the farmers, because they always ignore their health. They only think about the crop yield. But second, the farmers should be encouraged to regularly practice hygienic measures after spraying pesticides and use protective clothing and equipment irrespective of any inconvenience. Overall, extensive educational and training programs have to be initiated to improve the workers' perceptions on pesticides and attitude towards use of safe and hygienic measures during and after pesticide application. And I am stop here. Thank you everyone for quite listening and helping me when there are the technical issues.
Thank
Thank
you
you
so
so
much,
much.
Dr. Reena madam. It was a nice presentation and nicely done also. So ये technical चीज़ें तो चलती ही रहेगी as it is
Thank
in the
you.
online mode, so don't worry about it. But you are nicely presented. Thank you
Yeah.
so much. Thank you.
Thank you. Thank you, ma'am.
Next, Mayank sir.
Yes, madam.
मैम सर आप अपना पहले वीडियो ऑफ करिए बिकॉज योर वॉइस इज ब्रेकिंग.
Started breaking. Okay,
यस.
let me.
यस सर, मैंने आपको शेयर ऑप्शन दिया है. काइंडली शेयर योर स्क्रीन.
Okay. Am I audible?
सर योर वॉइस इज नॉट क्लियर. स्क्रीन शेयरिंग भी प्रोसेस में है. स्क्रीन शेयरिंग इज इन प्रोसेस. Hello. Sir, can you hear me? Mayank sir?
आप स्क्रीन शेयर कीजिए।
Hello.
Hello.
Already, yeah.
Participants, आपको मेरी आवाज आ रही है?
मैडम यस
Yes, ma'am.
That's why I'm saying
Yes, ma'am.
I will share from my side because it's taking
Hello.
lot of time.
Yes, ma'am.
So to save time in all the sessions, I will share from my side and you have to just explain. Mayank sir, can you hear me? Next, Sandeep sir, Sandeep Hatte.
यस मैडम आई एम
हाँ,
हियर।
yeah, we will start with your seminar.
ओके मैडम। हेलो
Hello.
मयांक सर
Ma'am,
आपकी वॉइस नहीं आ रही है, आप मोबाइल से ज्वाइन करिए। आई विल शेयर माय स्क्रीन फ्रॉम दिस साइड एंड यू हैव टू जस्ट एक्सप्लेन।
are you able to hear me now clearly?
नो सर,
Is
आपकी वॉइस
it
ब्रेक हो रही है।
audible?
योर वॉइस इज ब्रेकिंग।
Okay. Okay.
सो आई एम
So
स्टार्टिंग
I will
विथ
connect it from the laptop.
यस, आप मोबाइल से ज्वाइन करिए। तब तक मैं संदीप सर का प्रेजेंटेशन ले रही हूँ।
Sure, ma'am. Sure,
यस
ma'am. Okay.
यस संदीप सर, स्क्रीन दिसते हैं?
यस
कैन यू सी द स्क्रीन?
Yes.
यस मैडम।
हाँ, प्लीज
यस मैम
स्टार्ट।
इट इज विजिबल।
प्लीज
ओके
स्टार्ट।
ओके थैंक यू सो much madam. Dear friends, today's my topic of presentation is soil types, sampling method and its properties. I select this topic because I know all of you, most of the people I know that what type of background they are having. That's why I selected this topic, this particular topic. Okay. Myself, Dr. Sandeep Hate, I am Assistant Professor, Department of Botany, DRB Sindhu Mahavidyalaya, Nagpur. Ma'am, next please. So, dear friends, I am going to tell you about the introduction of soil, structure of soil, types of soil, sampling method, and its properties. Next, ma'am, please. Okay, as we all know that the health of soil is equally important as health of any person. We know that the soil is very important for us because it is more applicable when it comes to farming. But it is observed that nowadays health of soil doesn't get enough attention because people care more about the only cash crop and increasing quantity of the crop. As a result of this, soil become unproductive. So it's a need to awareness about the soil health should be increased to avoid this particular problem. Ma'am, next please. I hope I am audible, ma'am.
Yes
Am I
sir,
audible?
yes, everything is okay, please
Yes,
continue।
Okay, thank you, ma'am. So before going to discuss about the soil, we must understand ki what is exactly the definition of soil or what it contains. Soil, it is nothing but the mixture of mineral, organic matter, gases, liquid, and the countless organism that together support the life on the earth. Okay, it's a very simple definition. It is nothing but a combination of minerals, organic matter, gases, liquid, and different organisms. It is unaggregated or uncemented deposits of mineral. Okay, I think it is sufficient about the definition of it is nothing but the mineral matter, minerals, organic matter, gases, liquid, and the combination of the organism. Ma'am, next please. Okay, yeah, previous one. This is what I want to define you people, or I want to tell you about the particularly soil. Soil, it contain water, air, minerals, and organic matter. If you see the percentage of water, there is a 25 percentage of water, 25 percentage of air, 45 percentage of mineral matter, and only 5 percentage of organic matter. Okay, ma'am, next please. Yeah. What how this soil is formed? Before going to study the structure of soil, we must know the what how that process of formation of soil. Initially, you can see in this slide, the first one is the parent rock. That bedrock begin to disintegrate. The first one is going to disintegrate. Then in the second stage, you can see that there is a slightly deposition of organic matter, and in the third stage, there is a formation of different horizons. And the last one is there is a developed soil, which we will get it from the parent rock. Okay, I hope you are understanding the what I am saying. The process of formation of soil by the parent rock, we called it as a pedogenesis. But basically, if you see the pedogenesis, it is of different type. It may be physical weathering, it may be chemical weathering, or it may be a biological weathering. Most of the time, people, you might have read that there are only two types of weathering: physical weathering or chemical weathering. But there is a one more type, that is the biological weathering. It is due to the tree roots and the lichens. Okay, ma'am, next one please. It is nothing but the formation of soil. Then after the formation of soil, we are trying to understand what it contains. It contains different horizons, soil horizon. They are the layers of different types of soil found at different depth in the soil profile. Okay, and what is soil profile? It is nothing but a vertical section through the soil extend into the अनवेदर्ड पैरेंट मटेरियल. मतलब जो पैरेंट मटेरियल था, जो जिसका वेदरिंग नहीं हुआ है, तो लास्ट वाला जो पोर्शन है, दैट वी कॉल इट एज पैरेंट अनवेदर्ड पैरेंट मटेरियल. ये वाला जो सेक्शन होता है, उसको हम लोग बोलते हैं, वी कैन कॉल इट एज ए सॉइल प्रोफाइल. एंड दैट horizons, there are different types of horizons are there, A, B, C, and okay. Ma'am, next one please. Yeah, you can see here, as I told you that there are different types of horizon. The topsoil contain O horizons and A horizon, which is having an organic matter and leaf litter. It is again A horizon is also combination of this all these things. Then there is a subsoil is a combination of E horizon and B horizon, and the last one is parent material having a C and R horizon. This is the picture of soil profile. Okay, the next one please, ma'am. Yeah. uh, we are trying to understand the structure of soil. जो सॉइल प्रोफाइल का पिक्चर मैंने आपको अभी बताया था, जिसमें देयर आर डिफरेंट हॉराइजंस आपको दिख रहे थे, ए बी एंड ओ, ओ ए बी एंड सी एंड आर. ये जो सॉइल प्रोफाइल होता है, इट इज नथिंग बट अ कॉम्बिनेशन ऑफ डिफरेंट पार्टिकल्स ऑफ सॉइल. ओके, if you talk about the particle size of the soil, there are different particles. It may be a gravel, it may be a sand, it may be a silt, or it may be a clay. ये सारे जो particles होते हैं, they are going to form the structure of soil or the soil profile. Okay, ma'am, next please. If we see the size of the particle, then the basically the sand is having a less than 2 millimeter, silt is 0.5 mm to 2 mm, and clay is greater than 0.5 mm. Ma'am, next please. You can see here very clearly, clay is having a size of 0.002 mm, silt is 0.02 to 0.05 mm, very sand is 0.05 to 0.10 mm, fine sand 0.10 to 0.25 mm, medium sand to 0.25 to 0.50 mm, coarse sand 0.50 to 1 mm, 1.0 mm, and very coarse sand to 1.0 to 2.0 mm. Ma'am, next please. You can see here, you can easily we are easily able to differentiate what is sand, what is silt, and clay. I hope you all know that different types of soil. Okay, but if you talk about this particles of soil, they may be of different shape. There are different shapes of the particle. I'm not going into the detail of this particular, but simply I want to tell you the different types of particle. That is, they may be granular, they may be platy, they may be wedge shape, they may be subangular, they may be or angular, they may be prismatic or columnar. These are all the structure of the soil. Then types of soil. On the basis of they were present, matlab, on the basis of habitat, they may be of different type. Basically, there are 15 types of soil: black soil, red soil, laterite soil, desert soil, mountain soil, saline soil and alkaline soil, peaty and marshy soil, cohesive soil, residual soil, alluvial soil, marine soil, aeolian soil, loam soil, clay soil and glacial soil. Ma'am, next please. But some of them I am going to tell you about some of them. That the first one is, you can see black soil. We can easily identify the color of soil. I am again going to tell you how can we able to identify the color of soil in my next slide. Okay, this is black ones. Red soil is there. PT and marshy soil is there. Mountain soil is there. Ma'am, next please. Hello, ma'am. Next please. The saline and alkaline soil which we will get in and around the sea areas. Then the residual soil. Then last next one is a loam soil, and last is the clay which is naturally occurring in that. Here, listen very carefully. I told you about the definition of soil. It is nothing but the combination of organic matter and air and water and minerals. Okay. Then we studied that the structure of soil. The structure of soil may be seen that there are different horizons or the formation of soil from the parent rock to the fertile soil. Then we saw that the soil profile. In that soil profile, we seen that the different horizons are there, and after that we are trying to understand the what type of size of particles, what type of particle or particle is having what type of structure. They may be prismatic or angular or subangular. Here what we want to understand all the structure, because as I say that it is very important to all of us, because some of the may belong to microbiology, some may belong to the biochemistry or botany background or zoology background. We all know that soil is very important to all of us. If you want to study any type of floristic work or a faunal work, there is a definitely there is a role of soil is very important. That's why I select this particular topic to clear the idea about idea of you people. Okay. Soil sampling, it is commonly referred to the analysis of soil sample to determine the nutrient content, composition and other characteristics such as acidity or pH of the soil. Ma'am, next please. Yeah. If you want to collect the soil, we must have something with you, that is a field area to sample. We must know that what type of field area we need to sample, time of sampling, sampling tools, we need sampling tools, sampling that amount of sample, what type of what is the requirement of sample for the analysis of soil. We must know that sampling process, ड्राइंग सैंपल, सैंपल आईडी एंड शिपिंग, मतलब शिपिंग का मतलब है यहाँ पे देयर इज ट्रांसफर ऑफ सॉइल फ्रॉम दैट फील्ड टू द लैब कंडीशन. ओके मैम, नेक्स्ट प्लीज. ये सबसे इंपॉर्टेंट, दिस वन इज वेरी इंपॉर्टेंट. व्हेन एंड हाउ टू टेक अ सैंपल ऑफ अ soil sample must be taken at right time in the right way as described here, before planting anything in the field and after you taken off the crop. जब भी आपको सॉइल सैंपल को कलेक्ट करना है तो बिफोर प्लांटिंग या आफ्टर द हार्वेस्टिंग ऑफ द क्रॉप यू कैन कलेक्ट द सॉइल. बिफोर एप्लीकेशन ऑफ एनी ऑर्गेनिक और केमिकल फर्टिलाइजर यू कैन कलेक्ट द सॉइल. 3 months after application of any organic or chemical fertilizer, you must remember the thing if you want to go for the soil analysis. Then one more important thing, that is do not take soil sample from this particular area where the cattle graze under a tree and garbage area. You please keep it in mind. If you want to go for soil analysis, all this you please all remember this all this condition. Okay ma'am, next please. And how can we collect the soil? We need only 0.5 kg of soil required for the soil testing. And how can we collect? For soil testing, soil is taken from 15 different places. जो भी आपका फील्ड एरिया है उस फील्ड एरिया से आप डिफरेंट डिफरेंट लोकेलिटी से जिगजैग पैटर्न में सॉइल को कलेक्ट कर सकते हैं. डिग अ वी शेप पिट, आपको एक वी शेप पिट डिग करना पड़ेगा in the ground which is having a depth of 15 centimeter. It may depend on the what type of flora is available there. If you want to soil testing of the crop area, and it is the it depend on the its depth is only 15 centimeter. If you want to analyze the forest soil, it may extend up to the 15 to 30 centimeter. Please remember that. Follow the same procedure to take sample from 15 different places. Clean the sample by removing any garbage if present in the soil. Mix all the sample very well. Make a 4 equal parts of the mixed sample. Then mix two diagonally opposite parts with each other. Repeat the procedure 4 or 5 times, and out of that take only 0.5 kg of sample for the soil testing. I hope you understand. Ma'am, next please. Madam, next please. Priti madam. Okay. This is my team. I have studied that one village from Bangkok, that is a Bantha Vangsai village in Thailand. This one is my instruction. Ma'am, next please. Here I am going to collect that, how we'll collect that, right? Yes, this is very important. Soil color. Um, uh, this is caused because of the ह्यूमस कंटेंट. जो सॉइल का कलर, हमने जो 15 टाइप्स के जो सॉइल सैंपल्स देखे थे, इट डिपेंड ऑन द व्हाट टाइप ऑफ ह्यूमस कंटेंट्स आर देयर और आयरन कंपाउंड्स इन दैट पर्टिकुलर soil or area that define the color of the soil. Basic soil color indicate the indication of different soil types, indication of certain physical and chemical characteristics due to humus content and chemical nature of iron compound. Okay, this is the indication of soil. Ma'am, next please. Yeah.
Sir, your time is up. Kindly
Ma'am,
sum up.
last two slide.
Yeah.
Yeah. This is very important for you people. You can note down that Minsil soil color chart. You'll get it from the Google. I also collect this information from the Google. If you want to go for the identification of the soil color, you can use that Munsil soil color chart. It is available on the Google. Ma'am, next please. And the last one is the characteristics of soil is having a pH value. Ma'am, next please. pH is that is but as a measure of soil acidity or alkalinity. Ma'am, next please. Soil density and bulk density. Soil density is expected to, it is having a divided into two concepts, that is the bulk density and particle density. Particle density is the weight per unit volume of the solid portion of soil is called as the particle density, and bulk density is the oven dry weight of a unit volume of soil including of pore space is called as the bulk density. Ma'am, next please. Then soil porosity. The space occupied by air and water between the particle in a given volume of a soil are called as the soil porosity. This is all about the different characters or properties of soil, physical and chemical property. A chemical property, you can see that the clay minerals present, humus contents, cation exchange and soil reaction. These are the chemical properties of the soil. Ma'am, next please. And last one is the threats to soil, including the what type of threats soil are having. That is the first one is erosion, second one is contamination, third one is sealing, compaction, loss of biodiversity, salinization, floods, and landslide. This is all about the soil, its structure, and its properties. Thank you so much, madam, for
Thank you
giving
so
me
much.
this opportunity.
Thank you,
Thank you so
Dr.
much,
Sandeep sir. टॉपिक वॉज सिंपल बट वेरी इंफॉर्मेटिव. बेसिक था बट आपने अच्छे से एक्सप्लेन किया. थैंक यू सो मच.
madam.
डॉक्टर रेशमा मैडम.
Yes.
डॉक्टर रेशमा.
Yes, ma'am. Hello,
हाँ
am
हाँ
I
यस.
audible?
यस मैम यस.
There. Ma'am, cannot share. Okay. My.
No ma'am, you just explain from your side. I'll share the slides.
Okay.
Yes, please start. Can you see the PPT?
Yes, ma'am.
Yeah, please start.
All the UGC Human Resource Development of RTMNU Nagpur online refresher courses coordinator Dr. Dongarwar sirs, Dr. Priti Dharmik madam, myself Dr. Reshma Sonwalkar. Herewith I will present the seminar on the topic of phytochemical screening and evaluation of the fruit of Cucumis callosus for the mosquitocidal activities. Next slide. This is the abstract about this topic. In that the plant species selected from the Cucurbitaceae family. You know all about the Cucurbitaceae family, but these plants are Cucumis callosus. These are the very bitter in taste and some toxins are present in that plant. That's why I have selected these plants as a mosquitocidal activity. It is very valuable against the variety of disease, and in the present study, screening of phytochemical for the alkaloid, saponins, flavonoids, and tannins by the HPTLC method, especially use the fruit, because of the fruit are the very pulpy, juicy, and very toxic, or it is a very bitter in a taste. And for the two mosquito species are selected. One is the Culex punctifisciatus and Aedes aegypti. Main things in this my present investigation. Previous slides, ma'am, please. In my investigation, from the confirm that the fruits of the Cucumis callus is effective in mortality of the mosquito, and in the keywords is HPTLC, Cucumis callosus, and Aedes aegypti, and Cucumulus punctifasciatus, and cucurbitacin of I. It is the firstly reported from that plants. Next slide. Generally, we know about the mosquito are the very significant group from the insects, and which are the forms in the freshwater habitats, and they transfer the different types of the diseases like the parasitic as well as the viral diseases, in which include the filariasis, dengue, malaria, chikungunya, encephalitis, Japanese encephalitis, etc. So we have I have selected the two species, Aedes aegypti and the Cucumis. Sorry, Culex punctifasciatus. Aedes aegypti is responsible for the dengue diseases, and Culex is for the fevers. So in the plant species, Cucumis callus is a more valuable, in which the ripe fruit eaten raw as well, and it is used as a curries, green fruits, and it is used in the seeds are also used in the curries. Next slides. So materials and methods. The first methods is the preparation of the plant extract. So I have selected the fruit, and for the extraction, greenish mesocarp of the fruit, in which contain the juicy mild flavored tissue, and seed is carefully removed from the pericarp by using the spatula and shaken well to separate the yellowish fibrous part. That will be dried in a morses, and take a dried fruit into the 5 gram can be finely powdered using the stainless steel blender and extracted with the petroleum ether and methanol. That is the prepare the extract in a two chemical, that is the petroleum ether extract and methanol extract by with the help of the Soxhlet apparatus. Next. Next is the selection of the mosquito species. For the present investigation, two important species are selected, and larvicidal biopsy, that is for the biopsy stage, early 4-stage larva selected as a mosquito species is used. Experiment was conducted in the glass jar for the 24 hours, and 60 larvae can be exposing in a 5 concentration. One is for the controller, and these are triplicate in a form. 10 larvae in each. After 24 hours, recorded the number of the dead larvae and then reported the mortality comparing with the control. Next. Then another method is the HPTLC. So HPTLC, it is useful for the technique was followed for the qualitative analysis, and this done by the methods of the Passera analysis. So all the data are collected, and these data are analyzed by the one-way ANOVA method, that is the help of the SPSS software, that is the Duncan multiple range of the software. So result and discussion. So in my present investigation, it was found that the major toxicity observed in a 25 ml. This is the effect of the Cucumis callosus fruit extract against the Aedes, this species, mosquito species. So this is the control 5 ml, 10 ml, 15 ml, 20 ml, and 25. So most toxicity, both mosquito larva species, and major toxicity observed in the 25 ml. And sorry, previous slide, madam, please. These extracts were divided into two parts, methanol and petroleum ether extract. Methanol fruit extract was found greater mortality percentage in the 25 ml as compared to the 5 ml, 10 ml, 15 ml, and in a control mortality percentage was a nil. Next. And these results are discussed with the Govindarajan in 2010. Also observed the same result with the larvicidal and ovicidal activity of the crude hexane ethyl acetate, benzene, chloroform, and methanol extract of the 3 plants, that is the Eclipta alba, Cardiospermatum, Helicabum, and the Andrographis paniculata, which were tested against the early 3rd instar larva of the Anopheles stephensi. Next. And this is the result of the HPTLC technique. So with the help of the HPTLC technique from the Cucumis callosus, five constituents have been identified and confirmed from the fruit of the studied plants. So cucurbitacin I was identified and confirmed from the fruit of the cucurbitacin callosus. So main result is this plant is the cucurbitacin I was identified firstly. So result were compared with the experiment of the Riebger and Seifert and Sturms and Stuffner. In their investigator, cucurbitacin were found in citrullus colosynthesis, that is cucurbitacin E, glucosides, and minor amount of the glucosides of cucurbitacin I, K, L, and B. Next slide. And this is the HPTLC. Previous slide. This is the HPTLC image. And these plants actually, it is very useful for the farmer also, because they not require the high irrigations or very high nutrients or very high quality of the soil. These plants can be cultivated in a dry area, and these are not cultivated mostly. That's why the farmers can be. It is very useful for the side crop cultivate, and it can be cultivated these plants as a source of the mosquitocidals. Thank you, sir. Thank you, ma'am.
Madam, in conclusion, what you are going to say? What is the concluding part of your presentation?
So in my presentation, the Cucumis callosus cucurbitaceae family is selected because of it is a very bitter in a taste. So some toxics are present in that. So from this, I have find out the cucurbitacin I was responsible for the mosquito killing. And another conclusion is that it is very useful for the farmer for the cultivated, because of the they not require the very high irrigations, or it can be grow in a very dry area.
Is it a part of your PhD thesis?
Yeah, ma'am.
Okay, some part.
Some parts, some.
Okay. Okay, thank you.
Not total plant, not total parts, just a small part of my.
Okay.
And madam, this is the cucurbitacin I is a firstly reported from these plants.
Okay.
एक्चुअली ये जंगल के एरिया में ज्यादातर मिलते हैं, अपने महाराष्ट्र के बाहर इसे खाते भी है कहीं कहीं जगह में, जैसे कि राजस्थान वगैरह की जगह पे हो, तो कुकुरबिटेसी की कुछ वेसिस ऐसी है कि जो वेरी टॉक्सिक प्लांट है.
आपने कहाँ से कलेक्ट किया मैडम?
लेस हार्मफुल टू द ह्यूमन बींग्स.
आपने कहाँ से कलेक्ट किया था?
एक्चुअली ये प्लांट जो है मैंने सातारा जो है महाबलेश्वर, हाँ, और राजस्थान से भी इसे कलेक्ट किया था कुछ स्पेसीज.
Okay. Okay, ma'am. Thank you so much.
Okay.
Thank you.
Hello.
हेलो.
Hello. Yes, madam. I am here.
आपने अभी तक गूगल फॉर्म में इसे सबमिट क्यों नहीं किया था, या तो कल आपने
Madam,
क्यों नहीं मेल पे सबमिट किया?
Madam, because it was saying that the file size is
हाँ, so
limited.
you should ask me yesterday itself, na? On 11th
That's why
hour you are sending me on the email.
That's why I send it the PDF version of that particular presentation.
Where you have submitted? On mail or in
the
Google form?
Google Mail, I submitted the PDF version of our presentation because of the size limit issue.
On which mail ID? HRDC Nagpur 2020?
No, madam, your personal mail 05 Pritidharmik5 Gmail.
तो कुछ डेडलाइन वगैरह अगर कहीं पे मिलती है तो डेडलाइन के अंदर आपको सबमिट करना ही है ना सर? तो डेडलाइन
Yes,
का
madam,
कुछ
बस
इम्पोर्टेंस होता है कि नहीं? मुझे सभी
madam,
पार्टिसिपेंट्स को बोलना है। 1 मिनट, जस्ट मिनट, लेट मी फिनिश। मॉर्निंग तक सिर्फ 35 प्रेजेंटेशंस थे, उसके आगे के प्रेजेंटेशन आई विल नॉट डाउनलोड एट ऑल। मार्क एवरीवन, जितने भी प्रेजेंटेशंस आज आएंगे आई विल नॉट एक्सेप्ट दोज प्रेजेंटेशंस। जितने भी 38 39 जितने भी कल आए हुए हैं, ओनली उतने ही पार्टिसिपेंट सेमिनार अटेंड करेंगे, रातर सेमिनार प्रेजेंट करेंगे। रेस्ट ऑफ द पार्टिसिपेंट्स के पीपीटीज आई विल नॉट डाउनलोड, और सर आपके प्रेजेंटेशंस भी नहीं लेंगे। अगर कोई चीज़ की डेडलाइन होती है तो यू हैव टू फॉलो इट। कैन यू सी डॉक्टर मयांक? पीपीटी
yes,
दिख रहे
madam.
हैं आपको?
Yes, madam, it is visible.
सारी चीज़ें आपके मर्जी से नहीं चल सकती ना, कुछ रूल्स रेगुलेशंस होते हैं दैट यू हैव टू फॉलो।
Yes, madam, sorry to say, but I
नहीं, बाकी
tried to
पार्टिसिपेंट्स का,
upload.
दिस इज वन ऑफ द एग्जांपल्स, बट रेस्ट ऑफ द
Yes, madam.
पार्टिसिपेंट्स हु हैव नॉट येट सबमिटेड, they have to face problem regarding it. I am not going to download their presentations anymore. Yes, ma'am, please start.
Yes, madam. Ma'am, please play it in the. Yes, madam. Shall I start the
Yeah,
presentation?
please. Yeah.
Good afternoon, everyone present here. Myself, Dr. Mayank Varun, working as Assistant Professor of Botany in the Slub College. And the topic of my presentation is phytoremediation, the green way to clean environments. Next, please, madam. So rapid industrialization, anthropogenic activity, industrialization and urbanization during the last few decades have resulted in the accumulation of varied kinds of pollutant and different kinds of pollutions. Next, madam. Among the different kinds of pollution, soil pollution is of great concern, and particularly with the pollutant of heavy metals. Heavy metals are the metal which have a density greater than the 5 gram, and like they accumulate in the soil and causes the soil pollution. Various heavy metal pollutants, some of the examples of heavy metals are arsenic, cadmium, chromium, lead, and mercury. Next, ma'am. Now, what different sources of heavy metal pollution in the environments are mining activity, landfills, agriculture runoff, military operation, industrial waste, and refinery, etc. Next, madam. Next. Next. When these metals are bioaccumulated in the living organism, they show a toxicity symptom and causes different kinds of allergic reaction, diseases, neurological damage, and they even cause damage to the DNA and kidney and liver damage. Next. So two of the classical example of the bioaccumulation heavy metal in the human population are Minamata disease, which is caused by the accumulation of mercury, while a Thyroid disease, which is caused by the accumulation of cadmium, and both the diseases cause a neurodegenerative disorder and cause deformity in the human population. Next, please. Now, when these heavy metal emitted from the different sources, they get accumulated in our biosphere and different sediments. Now, the cleanup of these kinds of soil, sediment, and water body is a very challenging question to how to remediate these sites polluted with these kind of metal. Next. So this is the overall picture of the heavy metal polluted site identified all over the world. Next, madam. Now, to remediate this contaminated soil is a challenging question, and some of the techniques which are conventionally available to remediate these sites are physical methods, which include excavation and site disposal, chemical methods, which includes the treatment of the soil with the certain chemical, and thermometer. But all of these conventional remediation techniques which are available till now are expensive, time consuming, and they are further polluting the environment. Next, madam. The existing technology which are available till now are expensive, time consuming, and further degrading an environment, which force the scientists to come up with a new idea. And the scientists all over the world came up with a new idea of using plant to remediate these contaminated sites, and that phenomenon is known as phytoremediation. So phytoremediation is the use of plant and its associated microbes to remove, contain, or degrade the particular pollutant, specifically the organic and inorganic contaminants. Next, Mithun. So, in our nature, certain plants have been identified, which from the usual plant has the ability to uptake and accumulate heavy metals above the threshold level without showing the toxicity symptoms. These plants are known as hyperaccumulator, and the ability to accumulate these metal in the plant shoot or root part uh, used by the scientists to exploit the potential of these plants in the field of phytoremediation. Yes, next, madam, please. Till now, more than 450 plant species have been identified as a metal hyperaccumulator species. The first hyperaccumulator identified was Thalispi carolensis. Next, madam. So phytoremediation technology relies on the five basic processes: phyto extraction, where the inorganic contaminants are extracted by the root and then located to the harvestable portion. Next is phytodegradation. Phytodegradation technique in which the plant basically uptake the organic contaminant degraded into a less toxic form. Phytovolatilization is a process in which the volatile compound, organic compound, and metals, particularly mercury, arsenic, and selenium, are taken up by the plants and they are converted or volatilized into a less toxic form. Then phytostimulation is a technique in which the plant roots exudate certain chemical into the environment which stimulate the activity of bacteria which further degrade the contaminant present in the soil medium. And phytostabilization is another technique in which the plant root and plant itself excretes certain chemicals into the soil enzyme which makes the free available ion into a which reduces the mobility of the metals inside the soil so that it cannot leach down into the groundwater. Next, madam. So these are some of the plants which we have identified in a study conducted in the Firozabad region, which is particularly known for the glass industry. In our study, we have identified Terris vittata, which is a fun as an accumulator for the arsenic metal. Calotropis procera, जिसके हम आग भी बोलते हैं, is identified as a phytoremediated species for the cadmium copper. Likewise, Poa annua and Helianthus are identified for the phytoremediation of lead, copper, cadmium, arsenic, and Helianthus for chromium and copper. Next, Chenopodium album, जिसे हम भतुए के नाम से भी जान सकते हैं, which is a weed, is identified as a potential phytoremediator plant for the cadmium and copper, while Datura stromanium is identified as a potential phytoremediator for chromium, arsenic, and copper. Next, likewise, Withania somnifera, Eclipta alba, and Rumex dentatus are identified as a phytoremediator for copper, arsenic, chromium, respectively. So these plants which are found in the natural habitat are all weedy plant. Why we select the weed plant? Because weed plants are do not directly go into the food chain of the human population. In the aquatic stream, we found water hyacinth and uh, hyacinth as a potential phytoaccumulator for the metal silver, lead, cadmium, and zinc. Next. So for a plant to be identified as a ideal phytoremediator, the plant should possess a certain quality. It should have a tolerability or tolerance toward the metal for a particular metal. It should a high biomass and rapid growth rate. It should have possess a deep rooting system and it should be easily harvestable. Next, madam. So, advantages concerned with this particular technology, it is a green, clean technology, eco-friendly, time-consuming, and aesthetic pleasing technology. It is a sustainable technology, and it is used for the revegetation of the barren land, brownfields, and it is a low-cost approach, not consume too much labor and equipment intensive, and it utilizes the natural phenomena of the plant. It utilizes the natural phenomena of the plant, and sometimes it is also used for a biomining, in which the harvestable portion of the plant, after the harvest, metal is incinerated from that particular harvestable part. This is all for my presentation. Thank you all.
Thank you so much, Mayank sir. Nice
Thank you.
topic and nicely presented also. Thank you so much.
Thank you so much, brother.
So friends, here we have finished all the 4 seminars for today, and tomorrow we will continue with the rest of the seminars. Thank you all of you for today's session. Now you can leave the meeting and you please join that webinar link.
Madam, I want to ask you something.
Yeah, please.
Preeti madam.
Yes.
Madam, while we are just voicing a presentation, you are sharing the screen.